Okay, so this video is over chapter 22, the immune system. The immune system distinguishes between self and non-self, and they break it up, or they split it up into the innate immune system and acquired immune system. But there is some overlap, some intermingling between the two. So the innate immune system is general in nature, very general, or non-specific, whereas the acquired immune system is very specific. The innate immune system is present from birth. For example, your skin is part of the innate immune system. It is also based on inflammation. And uses this same strategy no matter the pathogen. It's not how you spell strategy. And what I mean by that, based on inflammation, is that it uses the process of inflammation to fight the pathogen. Whereas the acquired immune system is slower in nature because it is more specific, but therefore is more powerful. With the acquired immune system, we have the ability to acquire memory, and will build up over time. So we build up our acquired, I think your book might call it adaptive immunity over time. So let's start off by talking about inflammation. What are the main strategies for the innate immune system? So inflammation is caused by inflammatory cytokines. So we'll say inflammatory chemical cytokines. These are paracrine molecules. Meaning they act on neighboring cells or neighboring tissue. And the stimulus for inflammation would be like tissue damage that allows for, say, a pathogen like a bacteria to come in and cause or initiate the release of these chemical cytokines. So the purpose of inflammation would be to initiate or to provide a barrier bring in white blood cells, so immune cells, and also to promote healing. The 4 signs of inflammation are redness, heat, swelling, and pain. Okay, what we'll do is we'll group these two. So we'll put redness and heat together, and then we'll put swelling and pain together. Remember, heat does not equal fever in this instance. So heat is not the same as fever when it comes to inflammation. Okay, so make a note of that. So let's think about whenever we go running and our face gets red. Why does it get red? Remember, the blood is coming up and moving closer to the surface of our skin, essentially to release heat. And this is due to vasodilation. The same process occurs with inflammation. Now, swelling and pain is due to a number of things. The first is we end up with leaky capillaries. So our capillaries become more leaky to the fluid in our blood. And white blood cells can move out of the blood now into the surrounding tissue. This process the white blood cells moving from the blood to the tissue is called diapedesis. Diapedesis. And they do this by following these chemical cytokines. These chemical cytokines are called chemotaxins. And they create this sort of breadcrumb trail for these white blood cells so that they get to the damaged tissue, so they get to the right area. The other thing, remember, we have these leaky capillaries. Fluid is leaking out of our vessels. And with it, we get leaking of proteins, plasma proteins. And one of those plasma proteins are antibodies. Antibodies act like immune flags. So they go in, they attach to the antigen, the bad pathogen, and say, hey, look over here, white blood cells. This is something that's not supposed to be here. In addition, when proteins leave out of the blood, they also decrease osmotic pressure. And this leads to swelling in the tissue. So if plasma proteins are no longer inside the capillaries, inside of the blood, they no longer are pulling the fluid back into the blood vessel, and it gets left in the surrounding tissue. And the tissue begins to swell, and when something swells, it hurts. So that's how we get these 4 signs of inflammation: redness and heat, and swelling and pain. All right. Now let's discuss fever. Remember, fever is different than heat caused by inflammation. Fever is caused by a specific cytokine called pyrogen. And if you remember from lab, pyrogen is a chemical cytokine released by the granules of a neutrophil. So let's say you get infected by a pathogen, your neutrophils come in, they attack it, and they release this pyrogen from their granules. So what does this pyrogen do? Well, the pyrogen is released into the blood, and it travels to the thermostat of your body, and that happens to be the hypothalamus. The thermostat of your body. The hypothalamus keeps your temperature normally at 98.6 degrees Fahrenheit. But when we have the presence of these pathogens come in, what it does is it resets the set point. So pyrogens come in, it resets your set point. So now we have a new one. And let's say that we have enough of these pyrogens that our new set point is 102 degrees Fahrenheit. Okay? So pyrogens get released into the blood, they travel to the hypothalamus. The hypothalamus has these sensors for, say, for pyrogen, and whenever it senses this, it will accommodate to the situation and give you a new set point, and we set ours at 102 degrees Fahrenheit. Now, what would be a benefit to a new temperature set point? Well, we tend to work better. Our immune systems tend to work better or faster at a higher temperature. And some bacteria actually work worse at a higher temperature. Now, the cost of this new set point is that if it gets bad enough, if the infection is bad enough, and our set point just keeps on increasing, and say we get above 106 degrees Fahrenheit, we can actually end up with brain damage, possibly a coma, death, even seizures are common with an extremely high temperature. Okay. So continuing on with our innate immunity, let's talk about nonspecific phagocytosis. So this is carried out by a set of phagocytes, which include macrophages, eosinophils, neutrophils, and dendritic cells. So non-specific, meaning it is not specific to a specific antigen. Phagocytosis. So how do they know when to attack a pathogen? So let's draw an example here. Let's say that we have this pathogen right here. And he has this antigen on him. Okay? And your non-specific phagocytosis, let's say that, for instance, we have a macrophage here. It could be an eosinophil, a neutrophil, or a dendritic cell. And this is the receptor for the antigen on that pathogen. So how does it, if this receptor right here is non-specific, how does it recognize the pathogen, the antigen on the pathogen? Well, it recognizes a set of patterns so that it can be non-specific, and it can recognize patterns that are present in many different pathogens. And we call those PAMPs. And I believe it stands for pathogen associated molecular marker, something like that. So that would be a set of patterns that numerous different pathogens have in common. Okay, and the receptor responsible for recognizing these PAMPs is called a PRR, or pattern recognition receptor. Okay, so in our scenario, we've recognized the PAMP on this pathogen. So what happens within our phagocyte? Well, it engulfs the structure here. So engulfs it, or endocytoses it. So here it is. And it digests it, or breaks it down into different parts, and then it takes those parts and then posts them on an MHC1 molecule. Okay, this is an MHC molecule. Okay, that stands for major I'll spell that out for you. Major histocompatibility complex. Okay, MHC. We'll talk about two different classes of these. This one is an MHC1, and I'll draw that with one post right here. We'll talk about an MHC2 in a minute, and I'll draw that with two posts, so we know the difference. So what happened here? Our pathogen was recognized by our pattern recognition receptor. It was an endocytosed, digested by the phagocyte, and then a part of that antigen was then posted on the membrane of the phagocyte on an MHC1. So let's look at that in a little bit more detail here. So this right here is the same cell, and we're just going to look at this process in a little more detail. So let's draw ourselves a pathogen with a PAMP, and here is the pattern, the PRR, the pattern recognition receptor on our phagocyte. It engulfs or endocytoses it. It breaks it down for parts. It digests it. It sends it to the endoplasmic reticulum, which is holding these MHC1 empty platforms. Puts it on one of these MHC1 platforms. Sends it over to the Golgi apparatus, which then packages this whole thing right here, this MHC1 and antigen complex, and then exocytoses it to the membrane, the cell membrane of this phagocyte, and posts the whole thing on the surface. It says, hey, look what I found. So one thing you need to know about MHC1 molecules is that MHC1s are found on all nucleated cells. All nucleated cells have these. So we won't find MHC1 molecules on red blood cells. So that means all the cells in your body that have a nucleus will have MHC1s. So we also have the other innate immune cells called the natural killer cells, and they only make up about 2% of all your lymphocytes. Remember, lymphocytes are actually the smallest of your white blood cells. They identify these natural killer cells identify self cells. So cells that are yours that are supposed to be there. So they identify self cells that actually lack MHC1. So if they are supposed to have these MHC1 complexes and they don't, then these natural killer cells will come in and attack them. So what do they do to them? Well, they cause them to go through apoptosis, which is controlled, also called controlled cell suicide or death. Right? They do this by injecting in them granzymes, which are chemicals that sort of shut down things inside the cell, and also they secrete perforin, which makes the cell form holes. And what this does is it causes the cell to actually rupture, like with lysis, but instead of it exploding all its guts into the surrounding tissue, it has these granzymes that neatly packages it, and it's already shut down, so it's a neater death for the cell. Okay, so in addition, these natural killer cells, like I said, they identify self cells that lack MHC1s. Some viruses can actually come in and disable cells, MHC1s, and cancers can actually do this as well. That's how cancers will hide in the body. The last tool the innate immune system has that we're going to talk about has to fight against a pathogen is the complement system. And the complement system is a cascade of about 25 proteins. Okay. And we have a starting protein that ends up forming this complex called a MAC. Okay, like I said, it's a cascade, so we'll have the squirrely line that resembles or that's going to represent the cascade. Remember, a cascade is a waterfall of chemical reactions, one leading to the next and the next and the next. But I'm not going to ask you to memorize these 25 proteins. All right, so how does this begin? So we need a pathogen with its antigen here. And this pathogen is flagged by an antibody that has the specific variable sites for that antigen. So when this happens, when the antibody flags the pathogen, another binding site on the antibody will open up, sort of in the armpit of the antibody. And this binding site is called the complement binding site. So this right here is the complement binding site. And this is what is going to initiate this cascade of proteins. And what I mean by that is one protein's formed, and that causes the next one to be formed, and then the next one, and the next one, and the next one. Okay, so let's draw some proteins on here. And these are actually called opsonins. Opsonins. And the real definition for an opsonin is any chemical that can initiate phagocytosis. Okay. These opsonins are inflammatory molecules that will initiate phagocytosis. And at the end of this cascade, we formed this MAC. This MAC stands for, or MAC stands for membrane attack complex. And what it does is it pops the pathogen by forming a channel. So we have this sort of complex of proteins. So if each one of these is a protein, they formed this complex that then forms, say, a channel inside of this pathogen, and that causes the pathogen to pop. Okay? So we can see here how that maybe the innate system might have, well, we're going to see where we get these antibodies from, and then we'll see how the innate system also has a role to play with the acquired or adaptive immunity. And that has to do with how we get these antibodies. Now let's talk about acquired immunity or adaptive. This is broken up into two different parts, or into two different classes. It is either humoral or cell-mediated. If it's humoral, it is mediated by B cells or B lymphocytes. If it is cell-mediated, it is mediated by T cells or T lymphocytes. Both of these cells are born in the bone marrow, but mature, or I said go to school, in different places. B cells, B lymphocytes, will mature in the bone marrow, whereas T cells or T lymphocytes will mature in the thymus. Another thing that sets these two apart is that B cells will have different receptors from T cells. B cells, their receptors are antibodies. So if this is our B cell, its receptor will draw like this, with its variable region being specific for a specific antigen. Now the receptor for the T cell is simply called a T cell receptor. TCR for T cell receptor. So we'll draw that like this. And that receptor is also specific for a specific antigen. So you can imagine that if these cells are so specific and therefore have so much power, that they will have to go through some pretty rigorous selection. So what do I mean by selection? Well, their school, so their process of maturing, or maturation, maturing. So I have here three different categories: B cells, T cells, and then the middle one is when it applies to both B cells and T cells. So how do they mature? What is their school like? Well, first of all, they have to pick the correct receptor for their type of cell. So they have to pick a receptor. It's the first thing they have to do. They're still considered naive cells at this point. So they're not mature cells, not active cells yet. Then they go off to school. So B cells will go to the bone marrow, T cells will go to the thymus. Then they have to go through what's called negative selection. And notice that I'm writing this in the both category. That means both B cells and T cells will go through this. They both go through negative selection. And negative selection selects for cells that do not bind to self, meaning they're not going to attack self cells in your body that are supposed to be there. And if they pass this negative selection, then they will be termed, that'll be number 4. Then so if they pass negative selection, we can then call them self-tolerant. Self-tolerant. So know what negative selection is, and if they pass, what do we call those? So interestingly enough, that only 5% will pass negative selection. That means that 95% of these cells actually will have to die, and they do this by apoptosis. At this point, cells are still considered naive. They are not yet active, meaning they don't have a real function yet. So B cells are good. Now, what about T cells? Well, T cells, they still have to go through what's called positive selection. And positive selection selects cells that can bind to MHC molecules. And if these T cells pass, we can call them immunocompetent. Okay, so positive selection, if they pass, they're termed immunocompetent. So we'll learn that some of these cells need to bind to MHC2 molecules to become activated. Some need to bind to MHC1 to actually carry out their function. MHC2 molecules are only found on what's called an antigen presenting cell. Or an APC. APC, an antigen presenting cell. That means this cell is presenting an antigen on its surface. Not like a pathogen, but it's presenting an antigen on an MHC molecule on its surface, saying, hey, look what I found. That's an antigen presenting cell. These cells are B cells. B cells can be antigen presenting cells, dendritic cells, and macrophages. Can be antigen presenting cells. So know that list right there. Remember, MHC1s, these molecules are only found, MHC1 molecules are found on all nucleated cells. MHC2s are only found on antigen presenting cells. Now let's take these naive cells. We'll do B cells on this side and take them all the way down into when they become activated and into their actual functional cells. And then we'll do naive B cells on the left side and naive T cells on the right side. Okay. So what happens first? Let's do the humoral naive B side first. So the naive B side, naive B cell is a nucleated cell and has an antibody as its receptor. It's specific for a certain antigen, and lo and behold, it finds its antigen and binds to it. It engulfs this antigen and then will post it on an MHC2. So let's draw that on the next cell. Here's our naive B cell. It's still naive, not active yet. And here is our MHC2 molecule posting our antigen that we just engulfed. Okay? So we're not yet active. So let's see what it would take to activate this naive B cell. Alright. At this point, though, the naive B cell with that MHC2 molecule is considered an antigen-presenting cell. Alright, now on the other side, we have this naive T cell. The receptor for that, remember, is a T cell receptor. And it will be specific for a specific antigen. So we need an antigen-presenting cell. So let's do, this will be our antigen-presenting cell. It could be a dendritic cell or a macrophage. We'll do a dendritic cell. Okay. And it's presenting its antigen on an MHC2 platform, right? Okay. So our T cell receptor is specific for that antigen, so it recognizes it. And when it does that, it is now an activated T cell. And this activated T cell will become helper T cells, memory T cells, and killer or cytotoxic T cells. Okay, these helper T cells and these cytotoxic T cells, they also have another identifying molecule on them. Helper T cells also have a CD4 marker on them, and the killer cytotoxic T cells have a CD8. So make sure you know that it has that additional marker on it and which one goes with which. Okay. Now this helper T cell will actually go and be used to activate a naive B cell. So here's our helper T with its other identifier, or CD4, and it's going to have its T cell receptor, because remember, our active T cell still has its T cell receptor that's specific for that antigen. And if this T cell receptor is specific for that antigen, it will activate this naive B cell. And now we have an activated B cell. And now it can become its functional cells. It becomes plasma cells, which are basically factories for making antibodies. What type of antibodies? You got it. The same antibodies that were found on this naive B cell, the ones that are specific for that specific antigen. So it churns out all these antibodies, about 1,000 per second, or thousands per second, so tons and tons of antibodies. It will also become a memory B cell that, of course, has that same specific antibody on it. So the next time we see the same antigen, we can mount this response a lot quicker and produce more plasma and memory cells without having to go through the same activation process. So I think the last thing we need to talk about are these killer cytotoxic T cells. What do those guys do? Well, let's say we have this just regular old Joe cell here. And it's a nucleated cell, so it has an MHC1 on it. And that MHC1 is posting this antigen it found. This T cell receptor that's on this killer cytotoxic T cell is specific for that antigen and recognizes it on an MHC1 molecule. Remember, that is an MHC1 molecule found on all nucleated cells. It finds the antigen on the cell and then initiates apoptosis. Okay? So this cell then goes through apoptosis or controlled cell death. So what effect does the immune system have on homeostasis of the body? Well, it aims to push your body out of homeostasis. And it does this by changing set points in your body. Remember, set points are set point of regulated variables, like for temperature, body temperature, and blood glucose levels. It will change these for the benefit of the body so that it can work faster and better to rid your body of an infection. It will also change the sensitivity of targets, so targets of these set points, and the overall integrator. It will actually change the integrator that monitors and processes the information coming in that's maintaining homeostasis. Also, I ask that you know your immunoglobulins, your IgGs, which are antibodies. Okay, so know each of these and know the function of each of them. This is the most abundant in body fluids and protects against bacteria and viral infections. IgA right here is prevalent in mucous membranes, especially in the respiratory tract and the GI tract, also in saliva and in your tears. IgM is mainly found in the blood and the lymph, and actually is the first antibody made to fight a new infection. So it's the first soldier on the front. IgE is associated with allergic reactions and when your body is responding to allergens or antigens in the environment, such as pollen or pet dander. IgD is found in small amounts in the blood and is actually the least understood. They think it might have something to do with B lymphocytes or B lymphocyte receptors. So that was IgD. So the way I remember these is by remembering the acronym GAINed. So that lists all of them right there. The last thing I want to talk about is active versus passive immunity. So what is the difference between these two? Active immunity, we make memory. Passive, we don't get memory from. So what are some examples of these? Well, let's make a little chart here. With active, we need the exposure of an antigen. We have to be exposed to the antigen to form active immunity and get this memory. Passive immunity, we do not. Some examples of active immunity. I want to do natural active immunity and artificial. Artificial, we'll do that on the same thing on the other side. Natural and artificial passive immunity. So an example of natural active immunity is just simply having an infection. You're exposed to the antigen and you mount an immune response and you acquire that memory. Artificially, this would be like getting a vaccine. You're exposed to an antigen, part of a pathogen, and you mount an immune response and you acquire that memory. So that when you're exposed to it again, you can more quickly mount an immune response to fight off that pathogen. So what is passive natural immunity or natural passive immunity? An example of that would be receiving antibodies from mom, from, say, breast milk. There you're not exposed to the antigen, but you have the benefit of just receiving the antibodies that will be specific for that antigen, say, if you are exposed to it later on. But you did not develop these antibodies yourself because of an exposure. Therefore, you do not have memory, so you can't make more of these antibodies. Artificially, an example of this would be, say, like with a blood transfusion from somebody else, you could get transfused just antibodies in a serum or a fluid transfer. So we'll write transfer of serum, which is like the fluid containing the antibodies. Transfer of serum containing antibodies. So again, with this, these antibodies were not made by you, and it is not a natural process. These antibodies will have to be transferred to you by an injection, like if you were to get a blood transfusion. So that's an example of artificial passive immunity. There again, you would not be able to acquire memory because you were not initially exposed to the antigen. So of these two, passive would be less effective. Passive would be less effective because you are not mounting or not able to acquire that memory. But say if you are immunodeficient, like cancer patients that were not able to, if their immune system was compromised, this may be a applicable means of getting those antibodies. Of these two, which one is more transient in nature? That would also be passive. Okay, so this one is more effective. And lasts longer. Okay. All right, that's it for chapter 22. Happy studying.